Okay, guys, so now we're moving on into the adaptive immune system. This little lecture piece is going to deal with how the B lymphocytes work. But before we go into specifically the B lymphocytes, let's talk a little bit more about how the adaptive immune system works. Okay, so for this one right here, of all of these different types of immune cells, which ones are part of the adaptive immune system? Kind of gave it away. I already said B cells, but B cells are an example of a lymphocyte, and the other example of a lymphocyte is a T cell. So neutrophils and macrophages, those would be phagocytes that are part of the adaptive immune system, innate immune system. The two lymphocyte types are the T cells and the B cells, and all of those would be part of the adaptive immune system. All blood cells are produced in the bone marrow from pluripotent stem cells. So those lymphocytes would just be one of those blood cells that are produced from it. And then those lymphocytes then further differentiate into B cells and T cells. So let's talk about where those names come from. So the name comes from where they finish off that differentiation. For the B cells are going to be made in the bone marrow, but then they stay in the bone marrow and they become B cells in the bone marrow. So B stands for bone marrow. Whereas the T cells are also made initially in the bone marrow, but instead of staying in the bone marrow, they leave the bone marrow and they go to the thymus. Now the thymus is going to be sitting kind of right here in the top part of your chest. It kind of sits on top of the major blood vessels coming out of the heart. Okay, so that is the thymus there. And once they're in the thymus, they're going to finish the differentiation and they're going to become T cells. So the T is for thymus. B is for bone marrow, T is for thymus. Okay, so once they finish the differentiation, they're just going to start patrolling and moving around the body. And so they're going to leave their primary lymphoid organs, which is the B and the bone marrow and the thymus where they were initially made. And then they'll start patrolling around the secondary lymphoid organs. Okay, so what I mean by secondary lymphoid organs, those are things like tonsils in the throat area. Those are all of those lymph nodes, especially the ones found in the groin and the armpit and the neck. Okay, for example, if you can almost, sometimes you can see your swollen lymph nodes. Those are also things like Peyer's patches, which are found in the small intestine. And so what those lymphocytes are going to do is they're just going to patrol around. They're going to follow around the lymph nodes. They follow around between the tissues. They just sort of patrol around and hope, to look for something foreign, an antigen. Okay, so let's start with the B cells. B cells were made in the bone marrow and differentiated also in the bone marrow. When they're in the bone marrow, when they're differentiated, they start creating these little antibodies. So B cells have antibodies that they display on the surface of those B cells. And those antibodies have a particular shape. So antibodies are going to be specific to an antigen. Antibodies are a protein. They have this little special Y shape here. And those proteins create a very specific binding site that will only fit, again, like the lock and key model, a very specific antigen. Okay, so the antigens help distinguish self from non-self because they're going to bind to non-self antigens, things that are not part of your body. Let's look at the structure of antibodies. Antibodies are proteins. Those proteins are collectively called immunoglobulins because of the shape, and they're created from globulin subunits. And in those, they always have that very kind of specific little Y shape. And in that Y shape, you have two kind of pieces. You have the long chain here, which is in gray, and then you have the light chain, which is in green right here. And within the long chain and the light chain, at the very end of each chain, you have what's called a variable region, a variable region. And the reason it's called the variable region is because the rest of the antibody is going to be the same between all of the different antibodies because they're just the same protein. The only thing that's going to be different is the very end of that protein, because the end of the protein is what creates the binding site that is specific to the antigen. So in order for it to be specific, it has to have a very specific 3D shape that corresponds to the very specific 3D shape of that antigen that it's able to bind to. And so every single antibody that is created by each of those B cells, each B cell creates one specific antibody, and each antibody is going to be different from each other because they all have a different variable region that binds to a different antigen. Okay, so the variable region allows the antibodies to be specific to the antigen that they bind to. Okay, so this, remember when we talked about the adaptive immune system is specific. This is why. The antibodies are proteins that only bind to specific antigens. Okay, so let's talk about what happens when they wander around. This is called the clonal selection theory. And it's like most things that are named in anatomy and physiology, each word has a purpose. Okay, so let's talk about the first one. So in your body, your bone marrow is just spitting out all of these different B cells. And each B cell only makes one specific antibody. Okay, so in this one here, see A, B, C, and so forth, each of those are different B cells because each B cell makes one antibody, and all of the antibodies are slightly different from each other. So in your body, you've got this army of B cells that are just patrolling around, and each one is different from each other. Okay? So if each one is different from each other, that means that they have an antibody that is going to bind to a different antigen. And so what happens is if that B cell comes in contact with the antigen that is specific to itself, okay, so for example, in this scenario here, you got a bunch of different B cells, each slightly different from each other. B cell X just happens to come in contact with an antigen X that corresponds in shape to the antibody that it happens to express. Once that happens, it's like, hey, you know, I've been selected. I won the lottery because now I have found a pathogen that I can get rid of, and only me. Okay, so it will be selected to make clones. Okay, so what does the word clone mean? It means a bunch of copies that are all the same. So once this X1, which is specific and can only bind to this antigen X, once it binds to it, that selects this particular B cell to start creating clones of itself. That's why it's called the clonal selection theory. It's selected to make clones. And now what happens is your body is going to have, instead of just having one of that type X, just for example, type X B cell, it's going to make tons of them. Okay, it'll just explode the population of that one very specific one, which makes sense because if you have the pathogen that binds to that one, that is the only one where you actually need more of it. Okay, so it's going to create clones, and as it's creating copies of that one particular B cell, the second thing is it does one more final step of differentiation. Instead of starting at these B cells, those B cells then create two different types. Okay, so as it's dividing, it's going to change a little bit more. It's going to create two different types of B cells. The first are called plasma cells, and the reason they're called plasma cells is because those are the B cells that secrete antibodies into the plasma. Before this, the antibodies were only going to be found floating on the surface of the cell, so just sort of embedded in the membrane of the cell. But now what's going to happen is once it's been selected and it's making clones of it, some of them are going to start spitting out the antibodies, and then the antibodies are just going to be floating free in the plasma now. Okay, those are called plasma cells. And then the second type of B cells that will be created are called memory cells. What do you think the memory cells do? Their job is to continue patrolling around to make sure that whatever activated them in the first place does not come back. Okay, so when we say that the adaptive immune system has memory, the reason it has memory is because of the presence of these memory cells. Okay, so if we look at what happens with the immune system. So in this graph right here, along this axis, this is the number of antibodies that are found floating around in your blood plasma. This is just a representation of how active the adaptive immune system is at that point. On this axis here, we just have the number of weeks. Okay, so time period. So if we're looking at this graph right here, here is a blue line. This blue line represents the very first time that you are exposed to any particular antigen. Okay, so for example, somebody coughed in your face and now you've got the flu. And notice that it takes about 5 to 7 days for you to even respond to the flu virus. Okay, meanwhile, by the way, you're spreading it around to everybody else. Okay, so it's going to take about that much time to what we call incubate the virus. And what happens is during that time, you got to activate the B cells. You're undergoing clonal selection. You've selected the one that is specific to that particular strain of flu virus. And then what starts is after you do the clonal selection, some of them will be plasma cells, and those plasma cells are going to start spewing out all of this antibody. Okay, so we see a rise in antibody, and the antibody is going to then help take care of the flu virus and help get you better. Okay, so this just represents your first response to that specific strain of flu. Now let's say you get better and you go out again and somebody else coughs right in your face, and now you've got the same exact strain of flu virus again. So this pink line here, this pink line is representing your second exposure to the exact same flu virus. One thing that I'd like you to notice is not only do you get a much, much bigger response, lots and lots more plasma cells, but it happens much, much quicker. Within the very first week, within the first day, your body is already starting to fight it off. And because it's so quick and so big, often you don't even have any symptoms from that second exposure because your body is already primed and ready to get rid of that second exposure. This is why your adaptive immune system has memory. Those memory cells are already ready, and once they're here, they're going to undergo clonal selection again, create a bunch more plasma cells. Those plasma cells will start spewing out a bunch more antibodies, and that's going to help get rid of the infection very, very quickly. So quickly, often you don't even know that you got sick in the first place. Okay, so two kind of pieces here. Memory for sure, and also specificity. Please note that I kept saying the same specific flu cell, same specific flu strain. Okay, so keep that in mind. We can take advantage of the fact that our body creates memory for those specific pathogens by doing immunizations. And so what immunization means is you just take the thing that made you sick, the pathogen, and you kill it in some way. So you can do what's called attenuation, which means you destroy it, or sometimes you just take like a piece of it. And so you have something that could elicit a response. You then take that and you inject it into a person. And when you inject it into the person, that person is their body is going to respond to it. And it'll do the whole thing, do clonal selection, create antibodies, and that'll do the primary response. And along with those primary response, they're going to create those memory cells. And then once you have the memory cells, then they're going to patrol around. And that way, if you get sick for real, you'll have a bigger, much faster response, and you probably won't have any symptoms. But if you do, it'll be a lot more mild. Okay, so why do we need to get a flu shot every year? You have to get a flu shot every year because it's specific, right? So the flu shot that you got last year is for a different strain than you had it for this year. Okay, so you're going it only gets you, these memory cells are still specific to their antigen, and so they will only work for that one antigen. Okay. Oh, I wanted to mention that I usually get students ask, well, why doesn't it work? For one thing is it often does work by reducing the reducing the symptoms that you get. So sometimes it works just not 100%. And the other reason why a flu shot might not work is because, remember, within a flu shot, you're only getting immunization against specific strains, 1 or 2 or 3 different strains. So what they're doing is they're just sort of betting which strains are going to be the worst strains. There's actually lots of different flu strains every year. And especially if you think about within the different regions of the United States, right? There's all different regions. Each region is going to be hit with different types of strains. And so you might have an immunization against one strain, but you got a different strain, and that's why you still got sick. Okay, so what types of B cells secreted those antibodies? Remember, there's 2 different types. And so the ones that actually make the antibodies, those are the plasma cells. The second type are called the memory B cells. Okay, now we need to talk about, well, how do those antibodies work? After the plasma cells, here would be a plasma cell, secrete those antibodies into the bloodstream, into the plasma, into the liquid. How do they end up creating a response? Because the antibodies themselves are just proteins. They can't really do anything. So what they do is they work indirectly by getting the innate immune system involved. Okay, so they activate the complement proteins, which are part of the innate immune system because they are proteins that you will always have and don't change over time. And then once the complement proteins are activated, that is going to help get the innate immune system boost up, and so that they will then take care of the infection. Okay, so this is one of the bridges that I was talking about. Let's go into more, this is just a summary slide, so let's go into more specifically how the complement proteins activate that innate immune system. Okay, so remember, antibodies activate complement, complement then activates the immune system, and one of the ways they do that is by a process called oxidization, which just is a fancy way to say that they help the phagocytes bring the bacteria inside, the pathogen inside. So here's how it works. The antibodies, which are in red here, they're going to activate the complement proteins, which are these little green dots. And once the complement proteins are activated, they're going to create these complexes. And what that does is it helps the phagocyte, like neutrophil or a macrophage, something like that. It helps the phagocyte grab onto the thing it's going to engulf. Okay, so it helps with adhesion, and it helps grab onto it and then helps bring it in. Okay, so the process of opsonization just is a way to help or aid phagocytosis. And because the phagocytes themselves, it would be part of the innate immune system, those complement proteins are going to bridge adaptive, coming from the antibodies, to the innate, going to the phagocytes. Okay, that's the first way. The second way is by helping with inflammation. Okay, so once you have those antibodies. The antibodies can then activate the complement proteins. The complement proteins then activate the innate immune cells, things like eosinophils especially, and mast cells are shown here. And they're going to secrete cytokines. For example, a common cytokine is histamine, which you might know from antihistamines, which are the drugs that help prevent histamine release. Okay, and what the histamines do then is they make those capillary beds nice and leaky. That's going to draw in liquid, and along with the liquid, you're going to do extravasation, help recruit more innate immune cells to help clear up the infection. Okay. And then the third thing they can do is that those complement proteins can actually kill bacteria themselves, directly lyse the bacteria. And they do this by creating what's called a membrane attack complex. And all this is a series of proteins, okay? These are the complement proteins that insert themselves into the plasma membrane, which is shown here. So here's the plasma membrane of a bacterial cell. Those complement proteins are going to insert themselves into the bacterial cell, or sometimes host cells, your own cells that need to be killed because they're infected. And what it's going to do is it's going to make a big hole in the membrane. And what happens is when you make that big hole, that makes it so that the osmotic balance between inside of the cell and outside of the cell is going to be disrupted. And so what happens is once you make that big hole, because there's more solutes inside of a cell than outside of a cell, that allows a bunch of fluid to be sucked towards the solutes. Remember, solutes suck. Osmosis, there you go. Okay, so it's a solute suck, it'll bring all that fluid inside of that cell. And when you bring fluid inside of the cell, what's going to happen to the cell? It's going to pop. Okay, you just like overfills your water balloon, right? And so when you make a hole in it, it'll fill up with that liquid, and that's going to lyse or pop that bacteria or sometimes the infected host cell, and it'll kill it off. Okay. So, how do the complement proteins aid in the immune system? They help attract phagocytes. True. They form complexes that can puncture the walls of the pathogens. True. They link the innate and adaptive immune system. True. So, all of the above. Okay, on to the T cells.